Okay, so good afternoon once again. We have talked about the drug development and drug discovery phase. That was in our first session today. And we discussed about what is a drug molecule and what about the steps which we need to follow when we want to go for the development of a drug molecule. Okay, with that, I have told you about the definition of a drug, that what is a drug. Okay, so I discussed about that drug molecule is actually something that is designed just to go for the treatment of any disease. Okay, I talked about the drug molecule. That drug is a molecule used to treat a disease, but whenever you want to go with the construction of any drug molecule, so we need to consider any chemical compound or any compound that we are just extracting from any natural source. Okay, and it should have these features. Features includes that it should be safe, effective, stable, deliverable, available, and novel. So I told you that safe is it should be safe to our body without causing any kind of side effect or any other disease. Okay, or it should be effective to our body. Obviously it should reach the site of action, and it should be helpful for the treatment of the disease. For effectiveness, as in the percentage of effectiveness should be okay enough that it is helping for the disease treatment. Okay, stability refers to the property when we have talked about that a drug molecule should be able stable enough and should reach its site of action. It shouldn't be converted to some other alternate molecule or be broken up into its atoms. Okay, and deliverable, that is should reach the site of action. It shouldn't be that the drug is being consumed, taken inside, reaching the site of action, and it is just being excreted out. Availability is also important. The reason is that whenever the pharmaceutical companies they are going with the development of drug, the raw source should be available easily. Okay, and novel, obviously, because we were going with the designing of a new drug, a novel drug. So I told you that the study of drug is said to be pharmacology, divided into these two sub branches: pharmacodynamics, where we go with the study of the effect of drug on the body, and the study of how body reacts towards the drug, that comes under pharmacokinetics, talking about the admit properties, absorption, distribution, metabolism, excretion, and the toxicity. Okay, so all this is studied when we talk about how the drug is being acting inside the body, how it is participating in the metabolism of the cell. Okay, so what is the action of the drug, what is the mechanism of its action inside the cell, inside the body. So all this is included, all these properties are included in this whole process. But I told you that whenever we are going with the process of drug designing, drug development, basically, I discussed about drug development. So I told you that the first thing is we need to have a disease, and based on that disease, we are going to create a new drug. Okay, so disease should be there. Based on that disease, we are going to create a new drug, and the drug should go and bind at the target site wherever the disease cells are there, wherever whichever target site, whichever organ we are going to go with this binding thing, we need to search for a receptor site. Okay, because this will be the site where a drug molecule or ligand or a lead compound will go and bind and start its action. Okay, so the first thing was the disease should be there. Based on that particular disease, we will just search for the target site and the receptor site. We need to develop bioassays, that what is the process we'll just go forward to go with the in vitro and in vivo study. And then after this, okay, so after this we were going with the selection of the lead compounds. That was the first thing we need to do, was to select the lead compounds. I told you about there were various methods by which you can go with the selection of the lead compound, because this lead compound is actually the probable drug finally, which will help the in the treatment of the disease. So selection of lead compound was very important, that based on which method you are selecting your lead compound and how you need to go with the optimization of the lead compound. You can go with the construction of the analogs of a lead compound and optimize the properties, the structure of a lead compound. Okay, so all this comes under the computer aided drug designing, how you're modifying the structure, you're optimizing the structure of your lead compound. What about the structure of your receptor? Okay, so because we just can't go with the development, with the creation of a compound, a molecule which we are considering that this will act as a drug, this will go and bind at the receptor site and it will simply start its action. So this is not the way. First, the thing is we need to understand ki will this drug will go and bind to the receptor site. So this thing needs to be studied, and this we can't go for in vivo or in vitro study. We need to go for some kind of virtual study. We need to understand that if X is the structure of a drug molecule, then we need to have a Y site where it will go and bind, just like lock and key I told you earlier in the earlier session. Okay, so first we need to study the structures of both these things, the receptor as well as the drug molecule. And all this comes under our drug designing process, that is the computer aided drug designing. Computer aided just because we are using various softwares, various web-based tools in order to create the structure, in order to study the structure, in order to go with the study of the relationship between this drug molecule said to be the ligand molecule in CAD and the receptor. Okay, so when we are going with the study of computer aided drug designing, then we will call our drug molecule as the ligand molecule. It is termed as ligand. So whenever you go with the study, you will just study what is the interaction of ligand to receptor. Okay, so all this comes under the computer aided drug designing. I talked about the process of drug discovery, how the process starts the selection of the disease till the disease is there in the market. Okay, but throughout the process, wherever we are going with the selection of a target or the receptor, or we are going with the selection of a suitable lead compound, we need to apply the in silico study, the bioinformatics or the computer aided drug designing process to go with the structure prediction. Okay, like for example, if I am considering my target to be a protein, so we are very much aware that there are databases from where we can go with the structure extraction or retrieval of the structure of a proteins. Okay, so protein structures can be easily retrieved from the tertiary databases, like for example, protein databank. We can select the suitable receptor. If I consider that my targeted area is liver and there is a protein there, okay, I am considering the this is the protein X and I want to go for this protein as to be my receptor site. Okay, so considering that to be my receptor molecule, I will try to search for the structure of my protein in the database first of all. Okay, if I got the structure of the same protein in the database, then well and good. If not, then I can go with the creation of the three-dimensional structure. Maybe the protein which I'm considering is there on the receptor on the target side, that will be a suitable receptor molecule for me, is not there in the database. Okay, so what I need to do is I need to construct the structure. Okay, so in order to go with the construction of a three-dimensional structure of any protein, we are very much aware that we need to go for either the experiments, the experimental methods like X-ray crystallography or NMR spectroscopy, or in the first order, you can just move with the process of homology modeling or the in silico designing. Okay, so this can be the thing for going with the structure prediction of our target or the receptor. And the moment we have gone with the structure prediction, now we will go with the selection of a lead compounds. Lead compounds are those compounds which are having the suitable properties which will ultimately help us to treat the disease. So this will be the consideration while selecting a lead compound. So while selecting the lead compounds, we are just considering the properties. But in order to study the properties, I told you there is the structure of the compound, and we need to understand how these structures related to these properties and ultimately to the activity of the compound. So all this comes under the structure activity relationship model. We need to go for the construction of a QSAR model or a QSPR model. Okay, so while constructing a QSCR model, it will help us to understand the activity of the structure. We can go with the improvement of both the structure and activity if we are going with the QSCR model, because it is a kind of a relationship. It is a mathematical equation between the activity and the properties. So we are trying to understand how the compound will go and what about the properties of that particular compound. So initially we will go with this kind of a modeling process, QSCR modeling, and if we find that, for example, if we are having 100 properties, these are the number of properties, but the suitable ones are just might be 50 properties. So I need to improve those properties. How I can improve? I can just modify or optimize my lead structure. Okay, so based on this, we can go while constructing a QSCR model. There are various steps while going with the QSCR model, but the main purpose will be to just go with the improvement of the structure of any compound just before we are going with the actual construction or development of that lead compound. Okay, that will be a probable drug in future. So while selecting the lead compound, also somehow this in silico thing, this bioinformatics will work out with the construction, with the determination of the properties, and with the quantitative structure activity model thing. Okay, so the moment we have done with this, the compound which we have selected will be termed as a ligand compound. So ligand compound is something which we are going to study, and we need to understand this ligand is actually that molecule which will be the ultimate drug in future. So in case of when we are talking about the processes, the types of drug designing processes, so we can go with the two types of drug designing processes. The one is ligand based drug designing and the other one is structure based drug designing. Okay, so when we are going with the study of the lead compounds, I told you that lead compounds are compounds that are the probable drugs. We need to understand the structure, the activity of these compounds by the structure activity relationship model, and later on we'll just select the best of the compound and study the pharmacophore features of that particular compound. What are the features which the compound is having, and what about its interaction if it will go and bind to the receptor site. Okay, so all this will be studied, and when we go with the computer aided drug designing, we'll go with two methods. The one is said to be a ligand based drug designing, and the other one structure based. Ligand based just because the designing is dependent on the structure of a ligand, and it is said to be structure based just because we are going with the structure of a receptor. First we are studying the structure of the receptor. So what we are doing is we are just targeting the disease site. Okay, and based on the targeting of a disease site, we are going with the study of this disease site. Okay, so we are selecting our molecule, and the molecule is a receptor molecule. We are going to study the structure, the three-dimensional structure of a receptor or the binding site, and based on this structure we will try to search out for a suitable ligand compound. Or if we have already searched a ligand compound, okay, a ligand compound, that is a molecule, the drug molecule that will go and bind to the receptor site. So what we can do is we can just modify the structure of this molecule, the ligand molecule. Okay, so this will be the approach when you'll go with the SBDD process, the structure-based drug designing process, where what we need to do is we just need to go with the prediction of the structure of a receptor, and after the structure is being predicted, we have just analyzed the structure of our receptor site. We will go move forward with the structure of a ligand molecule. Whereas on the other hand, the ligand-based drug designing, what this is doing is that depending on the knowledge of the molecule which is binding at the target site. Okay, so first we will just go through the structure, the molecule for which we want to go with the construction of the ligand molecule. Okay, so based on that particular molecule, structure of that particular molecule, we will just go and search for a suitable receptor site. Okay, so ligand-based drug designing is when we have a defined lead compound, the structure is known to us, the ligand is there already, okay, and we have optimized the compound. So what we can do is we can search for a suitable receptor site. In certain cases, we can just go with the modification or the reconstruction of our receptor structure as well in these cases. Okay, so all this can be done on both the bases, like ligand based as well as our structure based drug designing, and they will just help us to go with the process of drug design. Okay, here you can see this is a small representation of how we can go with the SPDD process and the LBDD process. Okay, so both the methods, the ligand based drug designing process and the structure based, both will start with the first thing is we need to have a disease, and for that we will search for our target sites and we will go with the study of a lead compounds. Okay, so lead compounds and target sites, they can be of different natures. They can be protein, carbohydrate, lipid, or nucleic acids, I told you earlier. Okay, and based on this, we will go with the modeling of those structures. We need to go for modeling through the process of crystallography, X-ray crystallography, or we are going with NMR. Okay, and based on this, we need to study the structure and try to find out what are the possible active sites on that structure. Homology modeling can be done to go with the structure prediction process. And in order to go for the probable active sites, we need to study the pharmacophore study. Okay, so the pharmacophore of each molecule needs to be gone to study, and we will go through the structure on the whole and try to search out all the possible active sites where our molecule can go and bind. The lead compound, the ligand molecule can go and bind. Okay, so while going with the whole process, when designing thing comes, then structure of the receptor is designed first of all. And when we have gone with the structure based drug designing, then we need to go with the structure, with the study of the structure based pharmacophore. We need to understand how this structure will behave. What about its interaction when it will just go and interact with the ligand molecule? Okay, so when this structure and ligand, they are interacting with each other, the kind of interaction it is preparing, the conformation which is being prepared, that needs to be studied. And this is being studied by the process of docking. In the previous session, you have gone through the topic of docking as well. Okay, so in docking, what we are doing is we are studying the minimum energy conformation of these two molecules. So this receptor and ligand, they are binding like lock and key. They are preparing a conformation, and we need to have a minimum energy that is a stable conformation, a maximum stable conformation. Okay, so while going with the whole process, we will go with the calculation of energies. Okay, when we go with the energy calculation, basically there are two processes. The one like this homology modeling and all, where we are not considering any kind of where we are considering basically a template, and template based structure prediction is there. Studying of all the energies of the molecule is done in homology modeling. Okay, trying to create a stable structure, a minimum energy conformation, whereas we are not having any kind of template, and we are just calculating the total energy of the compound. Because if the compound is there, we are having these molecules, atoms are there, so what about the forces which every atom is applying on the other atom? Okay, so all these forces, these molecular dynamics under the molecular mechanics force field, we are going with the calculation of force field like hammer and charm. Okay, so there are certain calculations for energy calculations, calculating the scoring function and going with the process of docking. But on the whole, what docking is doing is it is just creating the stable conformation for your receptor and ligand. Okay, so we will go with the structure for the receptor and keep on the process trying to find out the most suitable docked ligand on that. So if we have found that this ligand is being binded on the receptor and creating a minimum energy conformation, then we will finalize, okay, this seems to be the best compound, the best ligand molecule, and we can just further move for the process of property determination for clinical trial phases and so on for the development of a drug molecule. Okay, so on the other hand, if we are going with the ligand based drug designing, where we are considering the ligand as the molecule which based on the structure of which we can just move ahead with the process of our designing, is the molecule which is supposed to be the lead compound, and this compound should have those suitable properties like oral bioavailability, selectivity, affinity, efficacy. Okay, and besides these properties, we need to understand what about the lipophilicity of that molecule, molar refractivity. There are various descriptors like TAF descriptors, steric descriptors. Okay, so there are a wide range of properties which is related to somehow with the atoms which are there involved in the compound, as well as whatever kind of interactive forces are there which are being developed between these atoms, between one molecule to the other molecule. Okay, so depending on this lead compound is being studied, the pharmacophore study is studied for this compound, and we can just go with the construction of the QSAR model again. Okay, so when we go with the study of QSAR model, then we will study the structure and activity, and we can keep on improving the structure with respect to the activity. Okay, if the activity of the molecule is there, we can just go with the improvement of the activity, and for improvement we can keep modifying the structure of a molecule. But whenever we are modifying the structure of the molecule, we need to consider that the structure should be a stable structure, a minimum energy conformation always, including all the suitable properties as well. Okay, after this if we are confirmed that while the molecule which we are considering as a ligand molecule, so what we can do is this ligand molecule should be a molecule that will satisfy all the properties. We need to understand how we can use this ligand molecule and study and predict the admit properties as well. Okay, so admit will help to understand that if the ligand is suitable enough, the structure is suitable enough, and will it go and bind to the receptor? Will it be able to absorb, or will it be able to distribute it across all the cells, moving across the cell membrane? Okay, based on the membrane potential, suitable membrane potential, following the Lipinski rule of 5. So will the compound be able to travel across the cell membranes? Okay, so the absorption, distribution, as well as what the compound will lead to, if it will lead to any kind of metabolic changes in the cell or body or not. Okay, so all this will be studied under the admit properties, considering that what amount of percentage is bioavailable, that is reaching at the target site, and what about the amount that is being excreted out from the body, and if it is leading to any kind of toxic effects. Okay, so all this will be there when we are going with the process of LBDD. Okay, so that was ligand based drug designing. Okay, so this is a small list. Okay, I'll be showing you these databases, and I'll be showing you there are certain drawing tools as well which are working for the construction for the designing process. Okay, so these are the databases: Zinc Database, CHEMBL, and JChem for Excel, Protein Databank, Binding Mode, and PDB Bind Stitch. Okay, the first thing is we need to have the structures. Okay, so these structures are the structures of our receptors. So like for example, if I'm having the structure of my receptor, a protein, so I can easily go to protein data bank. From there I can get the structure of my protein molecule. Okay, but on the other hand, if I want to go for the structure of any compound which I am considering, this can just behave and will help me to go for the treatment of the disease. Okay, the pharmaceutical companies, what they are doing, they are searching for all such kind of compounds. So they are going to the chemical compound library. Okay, and they are just going with the extraction or the retrieval of these structures or compounds from these libraries, from these databases. Or the other thing is, you can also go with the creation. If you have got through that this is a compound which I am considering, this is a novel compound, and I need to go for the structure prediction of this compound, then you can also go with the structure prediction, and you can also modify the structure with the thing which I told you, that you can go with the analogs of your lead compound. Like for example, you have just extracted a molecule, a compound from any of these databases, and what you're considering is that structure is not suitable and it will not bind to the receptor site, which I am actually selecting for the disease, for the target site. So what I can do is I can just modify the structure. So while during the modification of structure, I can just redefine the structure, keeping the structure to be an optimized structure. Okay, so this can be done using, there are various drawing tools. You can just draw the structures, you can study the structures. Okay, like ChemDraw is there, Marvin Sketch is there, ChemSketch is there, ChemWriter, UCF, Chimera, and PyMore. So they will just help you to draw these structures. Okay, so basically, in the process of drug development, the process of drug development starts with the disease, it ends with the drug molecule in the market. Okay, and in the whole process of, in the whole cycle of drug development, computer aided drug designing plays an important role. Okay, because when we are starting with the choosing of a disease, creating a bioassay, selecting the lead compounds, then after this we need to understand that we just can't select the lead compound and try to just move it to the market and let it be available for the patients. We need to just reconstruct its structure if the structure is not suitable to go and bind at the target site. So we need to have a suitable structure for that. So lead optimization is necessary, studying the interaction is necessary, and everything comes under computer aided drug design. It is okay. So it is actually a whole module itself. It will be very difficult to cover in 1 hour, because understanding about computer aided drug designing is, you need to understand what are the different processes, and what about docking. You need to understand what is molecular dynamics and molecular mechanics, going with the calculation of different types of force fields. Okay, and when you are constructing the structure, obviously you have to go with the study of its activity. So QSAR modeling itself takes a lot of time, because you are considering all the possible properties. Okay, when we go with the construction of a QSAR model, like for example, if I want to go with the construction of a 3D QSAR, okay, 3D QSAR as in, because we are considering various properties. Okay, how the structure looks like 3 dimension. So if I am considering a 3 dimensional structure, so not only I'll consider the atoms, the properties the atoms are having, okay, but also what about the various angles, bonds, and interactive forces which every atom is putting up on the other atom, or one molecule is forcing on the other molecule. So everything will be studied in that. And while going with the 3D QSAR, you need to understand that there are three scientists, this Hemet, Hansch, and Free Wilson, who gave their consideration with respect to certain properties. Like they have talked about lipophilicity, they have talked about the property of electronic properties, okay, and how the oral bioavailability, selectivity, efficacy, and binding, activation, response, all these properties are related to the compound and how it will act inside the body. It's talking about the admit properties. So all this will be covered in this computer aided drug design. The moment your ligand is ready to bind and docking is done, minimum energy conformation is done, then you can just go ahead with the process of your in vitro and in vivo study, going with preclinical and clinical trial phases to study that is it causing the right effect or not. Okay, so all this is about a brief about computer aided drug designing. I just talked only a brief about this. So I'll just move ahead with few of the practicals. Practicals as in, I'll just show you the. See, this one is something we have studied earlier also, because I told you that the selection of receptor is basically we can go with the selection of receptors like carbohydrates, lipids, proteins, and our nucleic acids as well. Okay, so proteins are the most suitable receptors for us, and we can just go with the selection of these proteins at the receptor molecules. Okay, so this is the most famous of all the protein data bank. Easily you can get the three-dimensional structure for your protein. Okay, so what you need to do, just need to type the name of the protein. Okay, just say I am considering, for example, keratin. Okay, my search here is just defined, just I have just mentioned the key term only. I'm not going with any kind of deep study for this keratin, because if you are just searching for a protein molecule or keratin molecule in terms of homo sapiens, or if you're aware about the accession or the id number for your particular protein, then you can just mention the name or id here. Okay, because if you're dealing with any such protein molecule which is already known to you, so you can just extract and retrieve the pdb structure here. We have already studied the structure, how we can go with the study of structure. Okay, so when we are going with the protein structure, you can easily go and download on the download files and download the structure here, pdb format. Okay, so when we are going with the structure prediction, so we can extract or retrieve from here, or if you want to go, we can go to Swiss model. I talked about Swiss model also in the process of homology modeling. So if you are not having the structure of protein as your receptor, if you're considering as a receptor in your protein data bank, you can easily go to your Swiss model, and you can go with the construction of your three-dimensional structure, or you can use modeler as well to go with the construction of the three-dimension structure for your protein. Okay, so this is the case where we are considering proteins. Okay, our receptors. Then we are having this, I'll show you, this is another database where you can just study about the chemical protein interaction networks. Okay, so here you can search for all the possible proteins. Here you can see there are by the name of the items, you can search for any gene, you can put the chemical structures. Okay, this is a format for chemical structures, protein sequences. You can for protein sequences, here you can see this is the amino acid sequences. You need to have that faster sequence for uploading the sequence, and you will get all the information with respect to the proteins. Okay, what about the sequences here, and how you can go with the structure extraction or retrieval. Okay, so for going with the prediction of the suitable receptor structure, you can use this database as well. Okay, besides this, we are having these two popular databases, both the databases Zinc and this CHEMBL. Okay, so they are helpful for the extraction or retrieval of our bioactive molecules or the compounds or the lead compounds. Okay, so both will help you. So here you can see zinc 5, it is very popular. You can just search for these substances or compounds. Here you can see this is I have clicked here in the substance. There is this catalog also. I'll just show you what it looks like. Okay, in the catalog thing, you can just browse for your okay, so all these are the different sources from where you can just go with the selection of any compounds. Like for example, I have just selected this one. Okay, so these are the sources from which you can retrieve your compound. Okay, so this is the source, this is actually maintaining the compounds, and this is having all these sorts of drugs. Okay, the drugs which are in man, they are there with these structures. You can go through the study. Like I'll just click one of the structure for my drug. Okay, so this drug molecule, this was added on this particular date, it is available. Okay, since this, what about the molecular weight? We can just go with the downloading of this structure as well. Okay, so there are various formats: smile format, SDF, CSV, XML, and JSON. So all these different formats are basically related to the different softwares or different analysis tools. Okay, what are all this? You need to go for a detailed study of one of the tools that we'll be doing in our last week practicals. Okay, so I'll show you what about all this. So here you can see this is the information with respect to the structure. Okay, I'll just show you here. This is saying you can go with the drawing of the tool, the structure. Okay, so this is the structure. This was the structure earlier, you can see here. Okay, and I can go with the modification of the structure as well. I can go with the modification of the structure. Okay, if I am selecting this C, I can just put C here. Nitrogen I can put. Okay, so you can just modify the structure, construct a structure here, and you can add whatever structures you want to add. If I want to add this benzene, I can add here. Okay, so Zinc database will help you to retrieve the structures for your compounds, and it will also help you to create a structure as well. Okay, so based on these structures, structure as in the compound which you are considering will be a suitable compound, you can construct it here. Okay, and based on this you can go with the study of whatever compound you want to go for. Structure addition of these structures or creation of these structures is just because possibly you are going with any kind of a novel compound or any compound which you are not aware what is the name of the compound, or you want to go for the analog for a lead compound. So both are the things which we are going to that is the reason you can go with the creation or the drawing of such compounds. Okay, the other database is this ML. Okay, so this is also a manually curated database, and you can have the molecules which are representing the properties of a suitable drug molecule. Okay, I'm just clicking here the search button. Okay, and you will be redirected to all the possible compounds, the targets, the essays, the documents about related to the drug molecule. Okay, so you can excuse me, so you can just see here, I just clicked here, there are these number of compounds, these number of targets. If I just go through the compounds and select the compounds, okay, so I can see these are the number of compounds. If I'm selecting one of the compound, this is a random search which I have done. Okay, I just clicked on the search button and I got to know that these are the number of compounds which are already there in the database. Okay, what is this compound? You can study this compound. The name of the compound is presently undefined. Okay, it is a small molecule. The molecular weight is here and the molecular formula is here. Okay, you can go with the structure search with respect to this molecule again. You are redirected to this is the structure. I told you the whole structure can be drawn. There are these sketches by which you can redefine, reconstruct the structure of your molecule. You can modify the structure of your molecule based on if you want to go with the analog of your lead compound, want to modify the structure based on the suitability that it will go and bind to the receptor site or not. Okay, so that can be done here. So the other one was I was talking about that how the molecules see chem sketch and this this although everything was embedded here only. अभी जो आपने देखा the sketching which we were doing was based on chem sketch only. Okay, I'll show you chem sketch also. Okay, so this is again a chemical drawing package only, and you can go with the creation of these chemical structures. Okay, you need to download it. The free version is there. You can go for academic license or commercial version as well. Okay, so obviously the three versions will be having a bit difference with the features. Okay, and you can go with the creation of the two-dimensional structures, the three-dimensional structures. Okay, and what about the properties with respect to these structures? So Chem Sketch is one of the sketching tool which is used when we are going with the study of computer-aided drug designing, and compounds can be easily sketched by using this. Besides this, ChemDraw is there. Okay, ChemDraw and Marvin Sketch is there. ChemWriter is there. Okay, I told you about all this. And then also I told you one of the name I mentioned was PyMol. Okay, this PyMol, RasMol, JMol, I told you about these all these softwares in the visualization study which I was talking about when I talked about protein structures. Okay, the protein tertiary structure prediction and visualization. So RasMol, PyMol, JMol, okay, all these softwares, they are not only helping in the visualization thing, but they are also helpful with the process where we can go with the creation or the structure sketching of these structures. Okay, and structures can be any molecule, it doesn't matter it is a protein or not. Okay, so it can be any molecule which is having a structure, it is having atoms connected through bonds, so it can be easily constructed there in your any of these softwares, any of these visualization, because not only just to visualize, they can create, they can modify, and you can study the interaction within the molecule, that how the molecule is working. But this is the case when we are talking about a single compound, a lead compound, or a drug molecule, or a receptor molecule. But then we need to understand what about the interaction between the two. So this Swiss dog, it is again a database, a tool basically, that basically helps to interact the study of ligand with the receptor, and particularly if we talk about the receptor to be a protein molecule. Okay, so all this is there. You can go through the study of his Swiss talk, where you will go through how the receptor is there and how the ligand molecule is working. Okay, so all this is again, you need to go for installation of this program. You can go for Swiss talk. There are certain other softwares as well, like Autodoc, which you can use. Okay, and you can go with the study, with the creation, with the study of the interaction and energy calculation, just in the manner that we have discussed in the Swiss model. Okay, this one of the softwares I'll just talk in my last session. Okay, and Argus Lab is actually helping us with the study of molecular modeling. Okay, so last session we talked about docking and molecular modeling, because the modeling of the structure is necessary when we are going with the modeling, with the study of its total free energy. Okay, the force fields which are being involved, then we need to go with the modeling of structure. We can go with the simulation of the structure, and Argus Lab is a suitable place where we can go with the study of these kinds of structures. Okay, so all this, these tools needs installation, downloading, installation, and you need to go with, first of all, with the basics. First you need to understand what is the structure, protein structure, and how you can go with homology modeling. First you have gone with the structure prediction process, then ultimately you can proceed with the process where you can go with the structure prediction of certain compounds which you are considering to be your lead compounds or the ligand molecules. Then after, you need to understand that there is a process called molecular modeling and docking, and how you can go with the energy calculation for the total conformation. Okay, so all this comes under the whole process. It is a very long process. It also needs a bit longer time to study all this molecular modeling, docking, QSAR, computer-aided drug designing, the whole process of drug development. Okay, so I just briefed you about the process, and if you're having any questions with respect to today's session, then you can ask me. Okay.